It is a very important concept, and it is a concept of technology, and today it is very important. And we have so many other things, like, say, for example, without medicines, some diseases cannot be cured with medicines, and those diseases can be cured genetically. And today we are going to see a few concepts of gene therapy and what is the meaning of gene therapy and how gene therapy is important, and how we can many diseases. So we'll see the slide. So what is gene therapy? It is an experimental technique that uses genes to treat or prevent diseases, and it can replace drugs. So I'd like to give you an introduction about gene therapy. Every organism is made up of cell, and the cell is made up of a storage unit which can transfer the genes from one generation to another generation, which is called as DNA. DNA is nothing but the deoxyribonucleic acid, and it is very important that this DNA transfers all the genetic material, and that is called as hereditary. And if we have to make some corrections in the hereditary or the basics of the gene, then we can cure many diseases. So this is an experimental technique that uses genes to treat or prevent diseases. So how it replaces drugs? First, let's see the basic structure of a DNA. So the DNA is made up of nucleic acid, nucleotide bases, and it is made up of purines and pyrimidines. It is made up of adenine, thymine, guanine, and cytosine. So the most important thing which we should know about, the basic thing which we should be aware of, is about the strands, and the most important thing is the DNA is very useful to make a protein, and we all know protein is very important for the body. That is why if there is a problem with the DNA, then naturally the protein which is formed in the body will not be a proper one. So hence, if there is a change in the structure of the DNA or in the components of the DNA, naturally the protein which is transcribed will not be a proper one. The simple concept is it is only because of the transcription of the mRNA. mRNA is nothing but the basics. I would like to tell you about the basics. mRNA is nothing but the messenger RNA. So the messenger RNA transcribes and then it forms a protein. Suppose if you have a structural change in the DNA, or in between the DNA you have some relocations or deletions, naturally there will be a problem, and the DNA will not be in a proper position to transcribe the protein, and it will lead to a lot of genetic effects. This is the basic concept of genes, and we should know about the genes before going in for a gene therapy. And to talk about genes, there are, see, every expression of a character in a man or a woman, it is all linked to genes. Example, if your gene is not expressed in a proper way, if it is expressed in an indefinite, say for example, improper way, naturally it will lead to this linked, gene linked diseases. Example, we can say cancer. So how we can eradicate this disease? It is possible with gene therapy, and it's going to create a great revolution in the world because scientists are undergoing and doing a lot of research on gene therapy, and they are using these genes to treat many diseases. And the most important thing is, as we all know from our primary education, we would have known something about the dominant gene and the recessive gene. We get one gene from the male parent, one gene from the female parent, and both fused together, we get an organism. And the expression of genes, I would like to state a few points about the expression of genes. The dominant gene, a gene which expresses itself and it masks the other gene. The gene which is not expressed is called as the recessive gene. So, example, if the father is tall and the mother is short, and the offspring is tall, it is because of the expression of the It is because of the expression of the genes. So when you're tall, the expression of the genes for tall, say for example, it is TT, it is expressed fully, and the other gene which is recessive, it is masked. So this is the concept behind genes and how genes are expressed. So I would like to state a few points on this. So expression, so everything, every activity we do, our kids do, it is all only through hereditary. So if a space is given for gene therapy, and it will really create a revolution in the world. So we are going to see a few methods involved in gene therapy. And one important thing is like about, I would like to explain you about what is a mutated gene. Before going in for the therapy, we should know the terms involved in gene therapy. So mutated gene is nothing but the transformed gene which is not in the proper position. Once the gene is mutated, either by deletion, by the concepts of deletion or translocation, naturally it will lead to an unhealthy copy. So, replacing a mutated gene that causes the disease with a healthy copy of the gene is called as gene therapy. So, practically, we may think whether it is possible or not. It is possible, and many scientists have done a lot of researches, and I have personally done some research on this. So it is a very important concept to know how to replace the gene, how to remove the defective gene and replace it with another gene, and what are all the target things needed for that. We are going to see a detailed description of gene therapy. And I would like to state an important point like other than, see, even when we think about the pharma industry, there are a lot of side effects in medicines. But in gene therapy, if we are doing it in a procedural way, in a very good method, using all the looking out for all the repercussions, correcting one by one, and doing in an in vivo way, then it will be successful. We have to remove the particular cell defective gene and then inject it with the proper one. So we are going to see the procedures and what are all the techniques involved in it. The second thing is about the method involved is inactivating the gene or knocking out a mutated gene. As I told you, mutated gene is a defective gene. It lacks one of the important part of the genetic material, or it is deleted or transcribed through various factors. It might be environmental factors or it might be genetic factors. So we are going to see two types of gene therapy, that is germline therapy and somatic therapy. And also I'm going to give you a few tips about how this therapy is carried out and what are all the procedures involved. So this is an important thing, introducing a new gene into the body to help fight a disease. So when someone is having a disease and suffering from a disease, how do we fight the disease? The normal way is taking a medication. And after taking the medication, for every medicine, there is a side effect. So if you need a permanent cure and that permanent cure stays with you continuously, then we have to insert a gene to fight a disease. So what is a gene? As I told you earlier, it is a physical and functional units of hereditary. And the genes are made up of DNA, that is deoxyribonucleic acid. And as I told you earlier, it is made up of purines and pyrimidines and adenine, thymine, guanine, cytosine, and nitrogenous bases. So every gene on a chromosome determines individual characters like eye, color, the way you think. So we are made up of genes and everything determines the genes. The activity which we do is determined by each and every gene. So these are the bases. And what is a gene mutation? So this is what we have to understand first before going in for gene therapy. So mutated gene is a defective gene. Suppose we have a genetic mutation, it clearly tells we have a problem from the hereditary, and I'm going to explain you what are the problems which we can face, hereditary problems. Say, for example, a normal gene is mutated. A normal gene has got some problem and it is translocated or deleted. Even one structure of the DNA is deleted or translocated, then it will really reflect in the generation. For example, it is cancer. We can take an example of cancer. Cancer is a very devastating disease. Hope we all know that it is only one of the reasons of cancer is through gene mutation. So when the mutated gene continues to stay in our body, it creates a drastic effect, and naturally, and the genetically modified gene will create repercussions and very dreadful diseases. So, in order to cure this gene mutation, we follow the therapy called as gene therapy. And gene therapy is a very important therapy because once we do it in a perfect way with certain modalities, the cure is permanent and we need not take any medications at all. So it is the permanent alteration in the DNA sequence. That's what I say. So it is permanently altered. And when the DNA sequence is altered, we insert something, we insert some corrected genes through it. I'm going to explain you what is a transgene. A transgene is inserted into the particular location of the defective genes. Hence, it replaces the genes and multiplies it, and the gene mutation is cured. So this is a very important concept. So for mutated gene, it is good to replace the gene using a gene therapy. So mutations can affect anywhere from a single DNA block, and it can affect the entire body also. That is why in cancerous cells, the duplication or the replication of the cells divide very faster and it spreads. So we are going to see the types of mutations. As I told you earlier, one is an hereditary mutation that is inherited from the parent, because in genetic combinations, when the sperm and the egg join or unite, when the embryo is formed, when there is a mutation or a change in the particular gene, it is called as an hereditary mutation. And hereditary mutation is actually bad because it is carried to generations and it is not curtailed to a particular generation. The next one, which I would like to tell you, is about the somatic mutations. Somatic mutations, it affects all other cells rather than the sperm and the egg. Say, for example, it affects the eyes, it affects the hair, it affects the blood, it affects the bone marrow. So, if a mutated cells affects all these places, if mutation occurs in all these places, it is called as a somatic mutation, and it is present only in certain cells. And somatic mutations can be influenced by the environmental factors like UV rays. Sometimes we say like a cancer has taken place in in a person. It's only because he worked in a place where there is more rays or cancerous agents are present. It is because of the somatic mutation. And hereditary mutation cannot take place because of the environmental factors. As I told you, it takes place only during the fertilization process. That is when once the egg and the sperm joins. And we all know this basic concept. The DNA copies itself during the cell division. So, and somatic mutations are not germline, as I told you. It affects all other parts except the sperm and the ovum. So we are going to see, this is a new term, de novo mutations. It is nothing but the new mutations. A new mutation is formed apart from the germline and somatic mutations. So we may be inquisitive to know what is this new mutation. This mutation will not be acquired from the parent, but a mutation occurs directly in the cell. When a mutation occurs directly in the cell, it is called as de novo mutation. So how it occurs in the cells, we are going to see the steps involved. So it is either hereditary nor somatic. And mutations occurs in the person's eggs or sperms, but this is uncertain. This is a medical thing which till today it is not determined. We do not know when the sperm and the egg combine at what juncture this mutation occurs. It is unpredictable, and this mutation will not be from the parents. Say, for example, if a mom and a dad is there, they do not have a cancer, but the child has a cancer. So I'm going to tell you something about transposons also in the future slides. So let's see a few things about this first. So it occurs in the sperm, person's egg or sperm, but we do not know the exact point it happens. So it is occurring in the fertilized egg shortly after the sperm and the egg unite. So even many scientists are doing the research till today, and we are not able to conclude at which point the mutation has occurred. And once the mutation occurs, it will be carried to the child, and the child will have a cancer, say, for example, leukoblastic anemia. So there are a lot of thalassemia, like a lot of defective genes because of this type of cancer. So, and this new mutation will not be in the parent, but it will be present in every cell of the child. So the child will go through all the symptoms of this this cancer. But it is not hereditary, as I told you earlier. It cannot be transcribed because only after the cell, the egg and the sperm unite, this de novo mutations occur. So, how do de novo mutations occur in somatic cells? So, we have seen in the germline cells, that is the egg and the sperm unite. The moment it gets united, a strong mutation occurs that is called as de novo mutations in a germline cell. We are going to see in somatic cells, as I told you, the bone marrow, the blood, all other things are called as somatic cells. So this happens in every cell in embryonic development, and it happens in the later stages, like when the embryo includes several cells. Hope you know about the mitosis, meiosis, the process of cell division. It happens during the cell division when more cells are added. So the cells which arise from the altered gene will have a mutation. So mutation is nothing but the alteration of the genes which is responsible for this cancerous growth. So let's see the procedure of how gene therapy works. So gene therapy is designed to introduce, as I told you, the genetic material we are going to introduce into a defective cell, and we have to make it into a beneficial protein. As I told you, it is all about the protein synthesis. The DNA and the mRNA transcribes, and finally the protein is formed. Hence, if the DNA is altered at any point of time, protein which is formed will be defective. That is the entire concept. And we are going to see something interesting and important in a gene therapy. So you may have a question in your mind how this process may happen. Because normally when we have a disease, we take a tablet and we find a cure for our disease. But here I'm talking about something insertion, taking out a gene and inserting a gene into the particular organism, how this all happens. And is it possible that directly we can insert a gene into an organism? No, of course. There we have to learn something about the vector. So vector is an important concept. Vector is nothing but the point which carries the transgenes. Transgenes is nothing but the guide. What do you mean by guide? It is nothing but the gene of interest. So what this gene of interest does is we are going to pick a desired gene which can cure the gene which can cure the mutated gene. So after taking this gene of interest, we cannot directly insert the gene of interest into the particular defective gene. Hence, we need a carrier, we need a vector. And this vector can be a virus, and we are going to see a few few types of vectors, and using that vector, we are inserting the wild gene or the good gene, which can replace the gene which is present, which is a defective, which can replace the defective gene. So how do we introduce the vector using a transgene? So we are going to see what are the different types of vectors. So as I told you, transgene is nothing but the exogenous gene transferred through vector. It is nothing but the wild good gene to replace the mutated gene. So what is a vector? A gene that is inserted directly into a cell, as I told you, usually does not function, and it is a carrier, and it is genetically transferred to deliver the gene. In simple words, I can explain you. Suppose we need something. I can tell you the example of Swiggy. So Swiggy acts as a vector. So they bring whatever we need to our house. So we do not get it directly. We have to go and pick all the things and prepare something in a kitchen for a food, for a good food. Instead of that, we order and connect the food to us through Swiggy. It is the same thing. The vector acts as a Swiggy connector to transfer the gene into the defective part. And we are going to see the what are the types of vectors. So viral vector, non-viral vector, and viruses, liposomes, and naked DNA are also access vectors. And I would like to tell you a point. Naturally, we will have a doubt in our mind if we transfer a viral DNA into the destined spot. What might happen? The first question which comes to our mind is whether the virus will affect the particular person, because we are having a vector as a virus itself. The thing is, before inserting this particular gene, this new wild gene, we take that virus, we inactivate the virus, and then insert that wild gene into the affected place. Then naturally, it will not produce a bad effect, and it will not have the following repercussions. And this step is very, very important in gene therapy. If a professional or a scientist who are not able to do it properly, it will lead to other side effects. Say, for example, if we are inserting a vector, a viral vector carrying a transgene into the particular affected place without inactivating the virus, there is a high probability that virus may affect that particular place, and it will lead to some other additional effects. So it is very important to consider this, and vectors are very important for transferring the transgene into the particular place. So we are going to see something about the gene therapy in somatic cells. So what is the, as I told you earlier, cells which are not involved in reproduction, the sperm and the eggs, except the cells of the sperm and the eggs, all other cells are called the somatic cells. And for carrying out the somatic gene therapy, we choose the bone marrow cells. I would like to tell you a important point regarding this. Why always scientists choose bone marrow? Even when they want to take a test in the somatic cell, they leave out other parts and they take a test from the bone marrow. There's a pivotal reason behind it. The bone marrow cells can divide and multiply in a very fast manner. So naturally, when a transgene is inserted into the bone marrow cells, what happens? The transgene enters into the bone marrow, and this host will destroy the virus cell. That is the virus vector. It destroys the virus vector, and it takes the wild gene. It takes the good gene, and once the multiplication occurs, it occurs drastically, and immediately the new gene will get multiplied. And that is how we replace the defective gene. And once the defective gene is replaced, then the result will be very good. And finally, the defective gene will be destroyed by the fast multiplication of this gene. This inherent ability is possible only with the bone marrow. That is why scientists choose bone marrow for this gene therapy. So it cannot be inherited, as I told you earlier. Somatic cells are not hereditary, so it cannot be inherited. And the success rate is highly noticeable in somatic cells. And two things I would like to tell you: we have to be very careful when we are doing this gene therapy in somatic cells or germline cells. And point number one is, when we are inserting the particular cell, we have to deactivate the virus. That is, deactivate the virus in the transgene. And the second thing which I would like to tell you, if you are inserting virus, this transgene, the vector, into a wrong place, it will lead to multiple problems. So these two things are important point to be considered. As far as somatic cells is concerned, we choose the target place as the bone marrow, because as I told you, varied reasons for why we choose bone marrow instead of other things. And even bloodline diseases can be cured using gene therapy in somatic cells, and only because of the division of the cell is more and it is rapid. It divides in a very faster manner. And the target cell will be the bone marrow cell, as I explained you earlier. So we have the limitations of the somatic cells, as I told you. This is the most important thing. If you're inserting the transgene into the wrong place, it may lead to varied side effects. And one more thing is, like, if the virus combines with the host or the genome, it may form a different thing. It is called as viral escape. It escapes and forms some other thing. So we have to be very careful, because it is all combinations, chemical combinations. And as I told you earlier, gene therapy, when done with pronouncement and perfection, will give you very good results. But if you miss at any point in between, it will lead to a lot of bad results. So doing it with pronouncement, having all the scientific equipments in a proper way matters a lot. And we should, again, the another point is, if we are growing a virus transgene, we have to be very careful. It should not be affected with some other viruses. And also, if a gene does not insert at the, as I told you earlier, I explained you that if you are not inserting in the proper location, it may disrupt the entire process. So the chances of activation of oncogenes. So we are going to see germline gene therapy. As from the word we understand, germline. So we understand it is with the sperm and the ovum. Some gene therapy which can be carried out with the sperm and the ovum is called as germline gene therapy. And the entire process will become a mess, and it will be reflected in the next generation, since it is a germline gene therapy. So before splitting the embryo, that is the gene of interest. COI is nothing but the gene of interest can be introduced into the germ cells. So what happens? Fertilized egg embryo. This method of transferring the gene is called as a germline gene therapy. But the point is, it gets to the next generation. So if there is a problem with us in choosing the vector or inserting the transgene into the vector, and then inserting the transgene into the inserting the vector into the target, if there is a flaw, this flaw will be replicated in the next generation. That is why scientists have a feeling that they are not more confident to conduct this germline gene therapy. So the changes made in the embryo affects the next generation. So in somatic gene therapy, this doesn't occur, and it is used for curing of many diseases. But in this germline gene therapy, there is a high possibility it can be a hereditary disease. It may create a hereditary disease, and for many generations, it may continue. That is why people do not go in for a germline gene therapy. This is the process about germline gene therapy. So cells of reproductive tissue are infected. As I told you, it is using a viral vector, and these modified cells are grown in the lab environment. So it shouldn't be infected. That's very important. It should be very careful, because as we see the picture, the result is a baby. And if the baby is healthy and all the process takes place in a proper way, there is no problem. Otherwise, this particular mistake will carry out for generations. So nowadays, people don't carry on with germline gene therapy. They are trying out with somatic. And so let's see what are all the techniques involved in gene therapy. So, see, genetics disorders are caused by loss of function mutation and abnormal gene expressions. And one important thing is like, as I told you, deletion. And this deletion is a concept, a part of the DNA gets deleted, and naturally, and sometimes the DNA gets replicated instead of transcribing in the proper way. So this concept of deletion and replication will lead to genetic problems, that is function mutations, that is abnormal gene expressions. It is nothing but the deletion. When it is deleted, there is an abnormal gene expression. So we are going to see the types of gene therapy. That is the first one is gene augmentation, and the second one is gene inhibition, and the third one is suicidal gene therapy. So gene augmentation, it is a very important process, and it is very interesting also. So the natural function of a gene is lost due to mutations. As I told you earlier, what is a mutation? The function of a gene is lost, and hence it cannot function on its own. Due to mutation, the normal functional protein will not be present, hence there will be a lot of altered reactions and consequences. So this mutant is replaced by fully functional wild gene. So how we can do it? It is like we can take it out. We can take the. We have to prepare a wild gene and then grow it in an environment in a lab, and then insert it with a vector. That will give a possible positive result, and hence it is translated with a protein. So this is gene augmentation. We are using it to treat monogenic disorders. So monogenic disorders, it is nothing but X-linked genes. Some disorders which happens in the X-linked genes can be treated using this gene augmentation therapy, and it is practically possible, and scientists have carried out this gene augmentation therapy. Gene inhibition, this is again a very nice concept. Sometimes what happens, the genes are expressed, it is nothing but the overexpression of genes. So for example, I think few people know about Mendel's concept of genes and how the genes are expressed. We know about the incomplete dominance, and how genes are partially expressed, and how the dominant gene suppresses the recessive gene. So all these are few things about the Mendelian's law, and few are applicable also. So here we see in gene inhibition, overexpression of the genes. The genes express themselves in a more manner. Say for example, I told you the DNA replicates, and there is another copy of DNA. This is a life-threatening situation, because there is going to be a great change in the methylation pattern and also the epigenetic profile of the particular gene. Naturally, it alters the entire combination and the nitrogenous bases, and hence the protein produced will not be the desired one. As I told you earlier, any change in the DNA structure will alter the protein produced, and hence a gene inhibition occurs. So the next is in gene, the overexpressed, that we know that we are going to introduce a gene using a vector and inhibit this overexpressed gene. So we are going to stop the overexpressed gene, and this is a very important method, and people follow this method for certain diseases. So this is by interfering the activity of the gene. That is another point. So as I told you earlier, there are different processes, like, say, for example, the transcription of the mRNA, that is messenger RNA. Then the last process is the formation of the protein. So, in between, if the process is hindered or inhibited, then the entire formation of the protein, the ultimate result we get will be a problem. So in between, if any of this process is overexpressed, we can insert our gene, that is nothing but the transgene which we have prepared, and inhibit the overexpressed gene. The third concept is suicidal gene therapy. This is again very important. It is what we can do is we can stop the growth of the cancerous cells by inserting the transgene, especially in the bone marrow. As I told you, the bone marrow has the ability to multiply the cells enormously, and hence it multiplies the cells enormously with more pronouncement. It can replace the cancerous cells. As we all know, there are a lot of for many types of cancer, there is no medication. So naturally the process is like inserting this particular transgene which we have prepared into that target place, and once it enters, this becomes a suicidal gene, and it is killed. It kills all the cancerous cells, the secondary growth cells. And once it is killing all the cells, finally after a particular period of time, this this transgene replaces the genes in the particular organism with the mutated gene. So you will not find a mutated gene after a particular stipulated period of time. That is called a suicidal gene therapy. And we all know this. Once the transgene replaces all the mutated genes, finally the result will be very good, that you will not find even a single cancerous cell. And if you don't find any cancerous cells, it may not get replicated. In few cases, these cells will be recessive. As I told you, these genes are recessive, and it will get expressed in some other generation. So we have to be very careful in carrying out a gene therapy. But as far as this somatic gene therapy is concerned, it will not be carried out to next generation or future generations. So future scope of gene therapy, I would like to tell you something about the transposons. So transposons is the very important concept which is going to be utilized in the future in gene therapy. So introduction of a transposons instead of the vector. So what is a transposons? It is nothing but a jumping gene. So a gene which can jump by itself, and it can modify its own structure, and it can restructure it, and it can be placed in different locations, is called as a transposons. So when we insert a transposon as a vector into the particular organism or the affected organism, mutated cell, naturally the gene therapy takes place in a very pronounced way. And right now there are a lot of researches going on this transposon particles instead of this other vectors, like as I told you earlier, the vectors of viruses and liposomes. Instead of using liposomes and viruses, this transposons has captured a very good place, and it is going to create a great revolution in gene therapy. And we are going to face a world, we are going to really see a world which is dominated by genetics, and genetics is really a miracle in the medical field. So there will be a scenario, or there will be a situation where even a person who is very defective, who hasn't got knowledge, who is called a dull-headed person, can be be made into a brilliant person only through gene therapy. It is not possible through any other medical mode. Like we cannot give any medicines. So any disease can be cured through gene therapy. But the probability is like we have to choose the correct one, the correct gene therapy with the correct procedure. It will give very good results. And I would like to tell about the in vivo and ex vivo. So how we can do it externally, if we are taking an organism or the transgene and we are introducing into the person directly to the particular affected gene in the place of the somatic cell, there is a high possibility this might fail. So it is better that we grow the particular gene in a very good atmosphere and in a laboratory, and then we inactivate the vectors, that is nothing but the virus. We inactivate the virus and insert the transgene, grow it into a proper way, and then insert it into the target, exactly into the target. That would be a proper thing. That would be a proper procedure rather than inserting directly into the person's somatic cells. So these are few procedures involved in gene therapy, and for sure in future we will have a lot of enhancement. And right now the scientists are doing great researches on transposons, and the only point is they are conducting a lot of research with mammalians and amphibians, and just carrying out this research with a human will take a little time, but surely only with gene therapy we can achieve permanent cure for all diseases. So I would like to tell you a few diseases which can be treated with gene therapy. The first disease is treating an eye disorder. This is a very well known thing, like how we can take out the mutant gene and replace it with a transgene, using all the wild genes and the positive genes and inculcating into the particular place. This is very popular also, and there are a lot of positive results in treating the eye disorders. The second one is cardiovascular diseases. As we all know, it is about the heart disease. And these diseases, see, why we have gene therapy, it is nothing but something which cannot be cured with medicine. It can be cured using a gene therapy. And I would like to restate the point, any genetic disorders can be treated only with the gene therapy. You cannot go in for a medication. Even if you go in for a medication, you will not have a permanent cure. So if you're looking out for a permanent cure, you have to go in for gene therapy. And the thing is, like choosing the proper vector and inserting it to the exact target place, these all matters a lot. And the next one is like hemophilia. As I told you earlier, somatic cells. If we are choosing the somatic cells for gene therapy, we choose only the blood cells and the bone marrow cells. Because the first reason, as I told you, the multiplication of the cells is enormous in these two places. And thalassemia also can be treated using gene therapy. Hemophilia is again a condition of the blood cells. So for any blood related diseases, we can use this particular gene therapy to cure. And any monogenic diseases. So monogenic diseases is nothing but the X-linked diseases. As we all know, what is an X chromosome? So any disease related to X-linked thing, it's called as a monogenic diseases. So any genetic disease which is present in our body, it can be used, it can be treated with gene therapy. I would use the word it can be, because the possibility is very high. Something which is corrected genetically cannot come again. That is the point behind it. And this gene therapy will not have any side effects once it is done in a proper way under very good medical conditions. If it is not done in a proper way or under proper medical conditions, there might be side effects. And as I told you earlier, there are a lot of side effects because of the viral infection. The virus, the vector in the virus in the vector may join with the host, and it may create a lot of problems. So it is always good that you do this particular gene gene therapy in a very good process. And thank you for watching this. Actually, there is a lot of things to be discussed in a gene therapy, and probably in the following sessions, I'll talk about transposons and the different types of vectors and what are all the future advancements and enhancements in gene therapy. Thank you all for watching.